The gram stain is not the only stain used in the identification of microorganisms of bacteria. There are other types of stain that we can use to identify microbes depending on the structure or what we are trying to identify. And another of such stain is the acid-fast stain. It's a special stain used for acid-fast organisms like the Mycobacterium tuberculosis. It involves the use of the Zell-Nelson stain, which is a differential stain that has two types of dye. The primary one is the carbon fuchsin, and the secondary one is malachite green or methylene blue, depending on the protocol. In the acid-fast stain, the acid-fast organisms usually retain the primary stain, which is the carbon fuchsin. So if you stain it and view it under the microscope, it appears red. You apply the primary stain, you decolorize with acid alcohol, then you apply the secondary stain. Acid-fast organisms usually retain the primary stain, so they will appear red. Another type we have is the endospore stain. In this, you are trying to view for spore-forming organisms like the Clostridium species or the Bacillus species. It's also a type of differential staining involving the use of two dyes. The first one, the primary dye, is the malachite green, and the secondary dye is a safranin. So endospores usually retain the primary color, which is the green color, when you view it under the microscope, while the vegetative cells usually retain the secondary color, which is a safranin, red, or pink under the microscope. Now we have another stain, is the negative staining, where you just stain the background. It's used for sensitive cells, are sensitive to dyes. So you stain the background and you place the cell there, in which the cell will appear transparent against a dark or stained background, so you can visualize it under the microscope. Then we have a simple stain where you just use one type of stain. You can use methylene blue. That we are trying to see just the arrangement or the shape of the cell. These are some of the other type of stains that we have. There are other types of stains, so you can tell us the one you remember.